Great. So it's over to Craig Liddicoat.
Thanks for the opportunity to speak today. So hopefully I'm not far away from finishing the PhD, but just to give you a bit of an overview of some of the research. Our group at Adelaide is basically looking into these links between biodiverse natural environments, their microbiomes, exposures for people and things living in those environments, how there might be changes to the host microbiome, and then flow on through to health outcomes. So there's obviously a lot of connections there, and it's a really multidisciplinary area. So we're just biting off bits and pieces and trying to build up the evidence base for that. So I want to try and stretch your thinking a bit today. So there's a lot of focus on how microbes and I've got a background in soils, and there's a little bit of focus on soil pollution and things like that. So there's a bit of focus on how microbes and soils and whatever can be bad for you, right? So I want to stretch your thinking a bit and maybe ask how those things can be good for you. So this image here is, we're doing this before lunch, so good timing. It's a traditional African hunter who's gone out and caught his organic free-range zebra. He's butchered it for lunch, and now he's washing his hands in the microbe-rich stomach contents of his zebra. So this is so far away from our concept of hygiene and public health that it's not funny. So, but I guess I'm sort of saying, or putting to you guys that from an evolutionary point of view, perhaps this is more than all the antibacterial sort of wipes and washes that we're using at the moment. So we've co-evolved with these microbes. We're building immune fitness. There's diversity of microbes in here that are competing with each other and perhaps keeping the pathogens under control. So, I'm not saying let's all go and wash our hands in poo, but it's a different concept just to sort of get your attention. So I'm just going to go back through some of those key concepts that Phil has laid out in a bit more detail. Give you some examples of some environmental epidemiology studies that I've done, and then get into some of the more mechanistic and more microbiome-focused stuff that we've looked at. So this concept of the biodiversity hypothesis, essentially what Phil's been talking about, where some immunologists and medical researchers have basically seen this decline in, so these are global biodiversity indices, sort of seeing these declines in global biodiversity, and at the same time this rise in your allergic, autoimmune and chronic inflammatory diseases. So this is the biodiversity hypothesis, that we're losing contact with this microbial diversity. A second sort of key concept in this space is the old friends mechanism. So this is more about your key species. So we had microbial diversity, and this is key species. So they can potentially come in from the environment, they can colonise, they can play a transient role, they can somehow modulate your immune system, both from a training point of view and an ongoing regulation point of view. And this old friends mechanism is come out of Graham Rook out of England. So there's at least 3 ways that microbes from the environment can help build your immune fitness. Number 1, so this image here is basically the lining of the gut, okay, as an example. These are cells from the gut lining, the gut wall. Microbes from the environment can come in and they can sort of supplement the protective microbial communities that are basically protecting your gut lining. So secondly, they can actively participate in immune signalling. So here we've got an immune cell on the reaching into the contents of the gut there, sensing microbes, bringing it back in, triggering a whole cascade of immune signals that can then influence the whole body. So this is stuff from your gut, and we're kind of learning that the gut is playing a central role in health. So this triggers signalling that can influence health outcomes right through your whole body. The third thing is that they're helping to build immune memory, your antibodies sort of scenario. Just to reiterate, we're talking about a biological connection between our environment and people, can influence the whole body, and through this immune signalling, it can influence both infectious disease, where you're triggering, let's attack those pathogens, and it's also influencing non-communicable diseases because these immune signals trigger tolerance, So we're talking about autoimmune and infectious disease where these things can influence. A couple of nature papers that kind of add to the story here. So, poor diets can cause loss So there's lifestyle habits that can cause loss of key gut microbes, and that can occur, you know, within a person and also through generations. Another key thing to think about here is that they found that your gut microbes are dominated by spore formers. So spore formers, you know, they activate in an anaerobic environment, but when they're out of the system, they form these resistant spores. They can persist, so they can, you know, they form a hard casing. They can persist in the environment, but then they can come back into an anaerobic environment and play a role. So there's things that can be out there in the environment that can come back into your anaerobic gut and do things. So just keep that in mind. Okay, so this is a conceptual dose response curve. You've got a key species, an old friend, maybe it's E. coli or salmonella or some sort of thing that provides a bit of immune training. So we've got low through to very high, and then obviously health response, bad, good. So too much E. coli, too much salmonella, bad, right? This is the type of conceptual relationship that, you know, we use in hospitals, and we're talking about immune-compromised people, but we're also trying to spread it everywhere. You know, the antibacterial wipes on the kitchen bench, the only good bacteria is the dead bacteria. This is what this is essentially saying. We think it's actually, this is how it should be in the real world, right? So obviously you've got, you know, too much of some of those key species is going to be really bad. But we're finding that, well, the thinking is that perhaps total loss of some of those species is causing this lack of immune training. Somewhere in the middle, you're getting this enhanced immune fitness from some level of key species and some sort of microbial diversity that keeps things in balance. Initially, the initial work in my PhD was asking the question, are these ideas supported in some of the large national data sets, the public health, the environment data sets that are out there? So a couple of papers here that I'll just touch on. Landscape biodiversity correlates with respiratory health in Australia. So area level data, looking at vegetation mapping classes across Australia, running a sort of a Shannon's diversity index for that across Australia, and then throwing in some other variables to just do a bit of a cross-sectional modelling approach. The second one here, ambient soil cation exchange capacity inversely associates with infectious and parasitic disease risk. So we're really looking at a proxy for soil microbial diversity and relating that to infectious disease risk. So the first one, basically, the most interesting results were sort of not for urban because I didn't have the best data for that, not for remote because I didn't have the best data. It was more this sort of regional Australia where my environmental covariates were suitable. Created a whole stack of potential explanatory variables, which is, you know, normally bad from a modelling point of view. But I used a machine learning version of multiple linear regression, I guess, which would chop out the sort of pick out the good stuff. And it's easy to interpret, which is why I kind of went with this. So essentially, you've got more hospital admissions for respiratory disease, less hospital admissions for respiratory disease, and these are standardised regression coefficients. So the bigger, the sort of more influence. So you've got how wealthy you are up the top and being beneficial, you know, as expected. Obesity on this side being bad and you know, an influence. Distance to coast was interesting as a, so the closer you are to the coast is better for respiratory disease, but up high and on the beneficial side were these vegetation diversity indices and species richness. And it wasn't just greenness or NDVI, they were down here somewhere. It was actually that quality of that green space that mattered. So, you know, this is just correlational. Next study. So this is talking about now interested in the soil microbial diversity, and again this is using an environmental mapping proxy for soil microbial diversity. And the thinking here is that if you've got diversity above the ground, you've got diverse root zone interactions and more complex habitat and feedstocks for the bugs below ground. So I grabbed an off-the-shelf, any soil scientists in the room? Okay, no? So there's an off-the-shelf sort of measure of soils, soil cation exchange capacity. So the amount of cations that are positively charged ions that a soil can store is kind of related to clay content and organic matter content. So, and both of those things generally provide diverse habitat for soil bugs. So I was using mapping of soil cation exchange capacity as a proxy for soil microbial diversity. And in the paper, I've got a copy of the paper here, found that that was supported, the use of that as a proxy indicator was supported in some of the biomes of Australian soil environments, microbial data sets that's out there for Australia. So, and I wanted to know, was exposure to soil microbial diversity perhaps building immune fitness, or were there better health outcomes? So this is a local government area data. We've got a breakdown by wealth, wealthy, middle and poorer areas, and then different soil classes. So the high, this is effectively high soil microbial diversity, so low soil microbial diversity. And in the poorer areas where you'd expect to see the results, we found that there was reduced disease risk in those better quality soils. So this is kind of supporting the general thinking that basically living in those areas with more microbial diversity from soils is giving you a health benefit. So attached to this, for the uncertainty modelers later on, in that paper I did a probabilistic sort of randomised pseudo individual level sampling to try and break up the area level data into more pseudo individual level data, and I ran some disease risk models and compared with soil and without soil, and the one with soil had a 7% better prediction of the disease outcomes in unseen areas. So soils are important. So I think I've got a reasonable amount of time still, I should go on a bit. So this is another paper, now getting into more of the environmental microbiome stuff. So what I did here was building on the study that Phil showed, where there was a restoration chronosequence in the Adelaide Hills. So we've got cleared, every dot here is a soil microbial community. So we've got cleared land, different years of revegetation, and then some remnants. So there's a bit of a flow there of restoration. And if you can read that little inset up the top, it's sort of stylized. The green arrow is the restoration. And then there's a couple of hundred sites from around Australia of soil microbial communities from, I've just put them into two groups, human altered or more natural. And you can kind of see a bit of we asked the question, is there perhaps a core microbiome that's changing here from disturbed through to more natural? So I'll jump straight to the summary thing here. Effectively what we found was that with ecological restoration, when you look into identified which bacteria were trending up and trending down along that gradient, and when you read the characteristics of the ones that were losing out when you restore, it was, these are fast growing, generalists, whatever, so opportunistic bacteria. And then the ones that were kind of building up more as you restored, they were slower growing and more finely tuned in terms of their requirements. So basically what we argued here in this paper was that the opportunists, and including potential pathogens, are increased in some of those disturbed soils. There's also potential use of this sort of a new way to look at the whole bacterial community as potentially a biological indicator of that system. So this is just using the bacteria that trended from one site in the Adelaide Hills and splitting up what are the ones that go down and what are the ones that go up. And then they kind of provide a pretty good differentiation for these other 200 sites across Australia. So there's and this is the opportunists versus the more niche adapters. So there could be a bit of use there for that. But when we looked at the human-associated bacteria, you kind of see, and this is kind of what happens in the Adelaide Hills, and this is what's happening across Australia, you see in these disturbed cases, you're getting things like at the genus level, Legionella, Enterobacter, Pseudomonas, Clostridium, which are, more or less opportunistic. You don't necessarily want to generalise that at the genus level, but we argued that there's potentially more of those potential pathogens out there in these disturbed landscapes. Hey, that might influence some of the susceptible people out there. So that was the argument there. And we have taken another step where we're trying to test these ideas experimentally and have run so this is taking out the view of the green through the window. This is taking soils, 3 treatments here in a mouse model, where we've got no soil, we've got soil from a low biodiversity area and soils from a high biodiversity area, putting them in a tray. We've got a little fan, and there's a light, the mice in the cage are getting a light dusting of this soil, so essentially it's sort of airborne. Just sort of testing this sort of ambient influence, because other studies have, thrown chunks of soil into a cage, they've injected things, they've force-fed them, so we're trying to go for a more natural exposure, I guess. This is not published yet, so sort of stay tuned, but just as a bit of a teaser, we found that the microbes, the soil microbial communities from these low and high, they were low and high macrodiversity, but they were also low and high microdiversity. We found that the gut microbiome of the mice was influenced by the treatments. We found that there were things in the soil that did transfer into the gut, and we found changes in the anxiety-like behaviour, and we think we've got a mechanism. So yes, stay tuned for that one. Just to summarise here, we think microbiomes can offer a plausible mechanism to link biodiverse green space with human health. As the data grows, we hope to be able to quantify that and put some dollars to it. The international biodiversity community want to mainstream biodiversity, obviously, into health, and they see microbiomes as part of that. And we think there's sort of opportunities for those win-wins. So.
Thank you very much.